So it's a real pleasure to be able to invite Professor Constantinidis to give us a talk today for our Systems Neuroscience and Complexity group. We're looking forward to hearing about some work where Professor Constantinidis has been stimulating the nucleus basalis, which is an area very close to many of our hearts, the main cholinergic input to the cerebral cortex. But this is in macaque monkeys, but importantly in the context of a working memory task. And so I think this actually provides a really important constraint because a lot of times when you look at brain stimulation experiments, they'll happen either at rest or even when an animal's anesthetized. And then you're really unsure of whether or not that stimulation effect is really impacting the normal functioning of the nervous system, or is rather just a signature of, let's say, its excitability or susceptibility. So I'm really excited by this direction that your work's taking, and really eager to hear more about the context and what you're thinking about in the broader scope. So thank you very much, and feel free to share your screen.
All right, wonderful. Hello, everybody. Good morning, everybody. I guess in Sydney is evening here in the U.S. So it's good to live in the future. Teleport in the future from time to time. They say don't live in the past. Well, I get to live in the future today. It is my great pleasure to be meeting your group today and getting a chance to talk a little bit about my work on the nucleus basalis stimulation. I know of all the great work that some of you have done, so I'll be very interested and very excited to hear questions and comments, and I welcome discussion. All right, so if I can now find the buttons in the new Zoom layout and share my own screen. Let's see. Second here. Okay, let me share my screen. All right, can you see my screen, and can you see the correct screen? Okay, very good. I see the thumbs up. All right, fantastic. So the topic of my talk today is working memory improvement through deep brain stimulation. And these are experiments that we do using a non-human primate model. And we use working memory as sort of the prototypical cognitive function, for which we know quite a bit, and we can use it as a model to understand the neural basis of cognitive functions and how neuromodulation, how a nucleus like the nucleus basalis or Maynard can increase or decrease or have an effect on these cognitive functions. Right, so let me just start by explaining how I understand the concept of working memory. So working memory in my definition is the ability to maintain and manipulate information in memory. And the classic example I like to give is, you go to a website to check your bank account, and you get a code for a two-factor authentication. This is the one-time code that flashes in your phone for a second, and then you need to maintain this information in mind for a few seconds to type in the website, and after that is no longer available. So what was the code? Exactly. Okay, so this was a 8-digit number, and that sort of reaches the capacity of working memory and the human ability. And the human brain has 100 billion neurons. But, we have to remember 8 digits for more than 10, 15 seconds, basically, we fail. So on one hand, this is sort of a real severe bottleneck of human cognition. On another hand, the fact that this exists allows us to read more easily the contents of working memory. So unlike long-term memory, working memory is very limited, and therefore we can read it out. And it's a tractable problem to try to find the neural correlates that maintain this information and read this out. So that is the advantage of studying a function like a working memory as a model of the neural basis of cognitive functions. And we use a non-human primate model in our experiments. Can you see my pointer? You can see my screen, but can you see my pointer?
Yes. Yep.
Very well. Great. So we use non-human primate. We're going to the trouble of using monkeys for our research because, number one, the monkey brain shown here on the right is a sort of a simplified version of the human brain. All the areas that have been implicated in working memory, like area 9, area 8B, area 946, all of these areas have homologs in the monkey brain, unlike, for example, the rodent brain that does not have this evolved lateral prefrontal cortex, and it's not the most natural model for studying a function such as working memory. All right. And we can train monkeys to do working memory tasks. So we don't quite show them do factor authentication codes, but we can train them to do a very simple working memory tasks, such the delayed response task that I'm showing here. Okay. So we can train monkeys to maintain fixation on a computer screen. We saw a visual stimulus somewhere in the screen, and then there's a delay period where only the fixation point is visible, and over that time period, the monkey has to remember where that stimulus appeared. And when the fixation point goes off, that's the signal for the monkey to tell us where that stimulus appears. And again, the monkeys unfortunately cannot communicate verbally, but they can move their eyes to this location of where they remember location of the stimulus, and again, tell us very accurately where they think that stimulus appeared. So this is a very simple paradigm. It has been used for half a century, but it's very powerful and really captures all of the essence of working memory. And we've used this paradigm along with other paradigms as a way to gain this understanding. All right, so why get in the trouble of doing these experiments in the monkey? The reason is that we can do invasive experiments in the monkey, so we can have microelectrodes advanced in the prefrontal cortex, and from the tip of this electrode or from the multiple contacts of these new generation of electrodes that are now available, we can isolate single neurons. So this is sort of a cartoon diagram, a simple diagram of one neuron in the prefrontal cortex of a monkey. As the monkey is doing precisely this delayed response task that I described, there are neurons that are tuned for the spatial location of the stimulus. So depending on where the cue appeared, to the right or to the top or to the left, the individual neurons show tuning for different locations of the stimulus, and therefore, and furthermore, they generate persistent discharges. So during the delay period of the task, when there is no actual stimulus in the screen, there are some neurons that continue to generate persistent discharges, and this delay period activity also exhibits tuning. Therefore, we have a neurocorrelate of working memory information maintained in memory. All right, so with this sort of very brief introduction, let me go on what I would like to discuss today. So first of all, I would like to take a sort of a deeper dive on what we understand about the mechanisms of working memory maintenance first. So the first part of my talk is not about acetylcholine, not about the nucleus basalis. It's just about the general mechanisms, the core mechanisms of how we understand, how we think that working memory is maintained. And I will speak particularly about this bump attractor model, which is, what we think the best model that we have now that maintains working memory. And only after I go that, then we can talk about these deep brain stimulation experiments and really put these findings that we have in the context of the mechanism of working memory. So if that's all right with everybody, I will start then with this sort of mechanism working memory maintenance. So how can a neuron generate persistent discharges? How can a neuron continue to fire action potential when there's no stimulus, no longer in the screen? We saw a stimulus, but then we turn it off, and over the delay period of several seconds, there are neurons that continue to fire action potentials. So it's actually not, this problem is actually not the most difficult problem in working memory. It is fairly simple to design a circuit that would maintain persistent discharges, as long as it relies on the principle of recurrent excitation. And this is sort of, I went too fast here. So this is a very simple model. Each triangle here is supposed to represent a single neuron. This neuron receives afferent input, which is ultimately driven by the external, the physical, the visual stimulus that appeared in the screen that drives this neuron. But this neuron also is interconnected with other neurons in the network, so that when the first neuron is activated, it will excite the second neuron, and then the second neuron will excite the first neuron, And this activity will reverberate through this pattern of recurrent connections, and this activity can be maintained. So in the context of the spatial working memory task that I described, we can think of neurons here as arranged in a ring. So each triangle now in this diagram represents a neuron with a different location preference. So this neuron would be maximally activated for a stimulus appearing at the 90-degree locations, which by convention that's the up position in the screen. This neuron would be maximally activated for a neuron at the 0 location. And as long as the strength of connections here, the footprint of synaptic connection depends on the preference of the neurons, we can achieve this recurrence in the network, and we can have activity be maintained in the network where even when nothing is happening. And this sort of model and this sort of network very easily explains also the inaccuracies of working memory. So why is it that we're not perfect in recalling exactly, these stimulus that we remembered basically, but we can mislocalize the stimulus, or we may forget a digit of the two-factor authentication. How does this bump attractor model basically explain that? And this is illustrated very schematically here in the bottom right diagram. So imagine that the stimulus appears at this top location, the 90-degree location. Now in this plot, the y-axis now is the preference of different neurons. So those are different neurons plotted along the y-axis. And we have essentially taken this ring here and we spread it along the y dimension of this axis. So if the stimulus appear at the 90-degree location, then the neurons that are mostly active are precisely these neurons whose location is 90 degrees. And we can have then a peak of activity in the network at this time point, and then we can track this activity over a period of time, over the entire delay period of this task. So this activity is maintained, this peak of activity or bump of activity is maintained in the network. However, due to stochastic factors, due to noise in the system, intrinsic noise in the system may drift, randomly over time. And at the end of the delay period, this peak of activity may not be exactly where we left it off, but in fact may have drifted, to the one direction or the other. And what the subject will recall then is not the original location, but it's exactly where the peak resides at the end of the delay period. And that, in fact, explains how the system can produce errors and can mislocalize. So this is what the model predicts. This is called the bump attractor model. And, it's a very simple model. It makes a lot of simplifications, but it sort of captures the essence of working together. Now, is that true? Is that true? So this model makes this prediction. Is there experimental evidence that in fact suggests that in fact activity does drift in the network, and in fact the behavior, what a subject recalls, really depends on this peak of the attractor. So those are experiments that we did about 10 years ago in collaboration with Albert Comte, he's been our partner in all of this, in a lot of these experiments that we'll talk about today. He's a computational neuroscientist in Barcelona, Spain. And so we analyzed activity of prefrontal neurons that were active during the delay period. And we said that, if it's true that when the bump of activity drifts in one direction to the left of the real location or to the right, here I have rotated these plots that I saw in the previous diagram. Now the x-axis here is stimulus, the y-axis is time. So if it's true that this bump activity sometimes drift to the left and sometimes drift to the right, then we're expecting these leftward drift trials, the subject will make an error slightly to the left of the reallocation of the stimulus. In these trials where the bump of activity has drifted to the right, we would expect the subject to make an error on the other direction. So we did this analysis backwards. We took the actual endpoint, the actual behavioral output. We saw where the monkeys made eye movements where the endpoint of saccades terminated, basically. And we broke down those that are terminated slightly clockwise or counterclockwise relative to the mean position of its stimulus. And then we plotted the tuning curve of firing rates drawn from these left and rightwards, basically, deviations. And in fact, we saw that this peak of activity differs systematically. So this is just a single neuron example, and it's very subtle, but over hundreds of neurons, basically, we saw that this was very consistent and very systematic, and there was, in fact, a difference, basically. I should say this finding, is not trivial. This agrees with the idea of an attractor model that has drift left and right, basically. But this prediction would not be borne by a model that decays. So if we assume that the inaccuracy, the errors of working memory are due to a decrease in the amplitude of this firing rate over time, this second model does not make a prediction that this tuning bias will change over time. And this is plotted over here, the tuning bias as a function of time during the delay period of a working memory task. The bump attractor model predicts that this increases monotonically as with time. If we had decay, however, this would not happen. It would only emerge very instantaneously at the very end of the trial, and the experimental data unequivocally show that we had this buildup of tuning curve bias. All right, I want to show one more slide, in this part of my talk, basically, and talk a little bit about activity silent mechanisms. So in recent years, there has been a lot of discussion about alternative mechanisms. So I presented you these results of persistent activity of neurons generating spikes during the delay period of the task. But there's evidence now that there are other mechanisms that also play a role in the maintenance of working memory that may not necessarily depend on covert, on obvious increases in firing rates. And these are generally called activity silent mechanisms. So one such phenomenon is the phenomenon of serial bias. So if you have to remember where a stimulus appeared in a particular trial, the kind of errors that subjects do, monkeys and humans alike, depend not only on the position of this, the present stimulus, but in fact, the position of the previous stimulus. So this is in a way is an inaccuracy of working memory. If working memory were perfect, then once you've seen a stimulus and you've made a response, and you finish the trial, you should completely wipe off this previous memory, basically, and not have it influence any subsequent trial. But in fact, the human memory and the monkey memory does depend on this previous stimuli. So if the previous stimulus that is no longer relevant, has absolutely no meaning, no significance for the current trial, if the previous location were here, there seems to be some residual trace, basically, of this previous stimulus that influences the current memory. And in fact, subjects tend to mislocalize this last stimulus that they saw in the direction of the previous stimulus, particularly for certain orientations, certain relationships. If the two last stimuli essentially are fairly close with each other, then you have an overall attractive effect. And that's shown over here. This is sort of the curve that shows the error of the current trial as a function of the location of the previous trial. All right, now why we got interested in the activity in the serial bias? The reason we did is that when we looked at the activity of neurons now in the interval between two trials, we saw that after the first trial has ended somewhere over here, the fixation of one trial has gone off. Basically, the monkey makes an eye movement, receives reward, there's an inter-trial interval, and then the fixation point turns on for the second trial to begin. There's at this point, there is no trace in the firing rate of the neurons. If we try to decode the previous stimulus is no longer present. And this is shown over here. So this is the tuning curve for the previous stimulus during the delay period of the trial as it still goes on. this is what happens right after the monkey makes a response, and we see that kind of inverted, because the monkey is making eye movement, and now it has to go back to the fixation point. But at this point in time, this has been wiped out. So the firing rate, the spiking of the neurons, no longer represents what happened in the previous trial at this time point. However, right before the next stimulus appears, we saw this trace be reactivated, and this yellow line here, this orange line, is exactly at the end of the fixation interval before the next stimulus appears. And without going into excruciating detail here, I mean, in essence, what this shows basically is that after the new cue appears, the new stimulus appears, we have a reactivation of the previous trace, and it is the two bumps of activity that merge together that in fact pull this activity basically of the new stimulus in the direction of the previous stimulus. All right. So the point I want to make here is that even for this activity silent mechanisms, which we see and we believe, and they're real, and they don't depend on persistent activity, the bump attractor model still explains what happens and what is it basically that how the error basically appears in behavior as a result of this merging extension of these two bumps of activity. So for any way that this activity was able to be maintained and be reactivated, it is the spiking activity at this time point that ultimately is going to generate behavior. All right, so let me pause then here and summarize this first part of my talk. So neurons in the prefrontal cortex maintain discharges through recurrent connections. Activity drifts with time account for behavior, and activity silent mechanisms are real, are evident, but they influence behavior in as much as they influence spike generation. All right, let me pause here before I move on and take any questions, if there are any questions so far.
So I was curious what you would consider the kind of competing models for a bump attractor model. I know there's we were chatting a couple weeks ago with in the lab about David Susselow's recent paper looking analyzing Robert Yang's RNN work, which some of the tasks that they train have a working memory-like component, and there you think about the distance to a particular fixed point in the dynamical regime of the network as maybe something like the working memory capacity. And I was just curious kind of how you view that versus other types of models.
That's a very good question. So the RNN models or some RNN models do not necessarily generate persistent activity. So they're not active during the entire time. It is short-term plasticity of synapses. So similar to short-term facilitation or short-term depression, basically, of synapses that allows a neuron basically to fire differently, after one stimulus relative to the other. So that I would consider that sort of equivalent of this activity silent mechanism. So one class of model basically allows for working memory to be maintained without continuous generation of persistent, action potential generation. And I think there's definitely evidence for that. Now, the second class of model for which I didn't speak at all, in the interest of time, but I'll be happy to discuss as well, are models that depend on rhythmic activity. So these models care not only about neurons generating discharges, but exactly the phase and frequency of stimulation. And the models proposed by Eli Muller and colleagues, for example, rely on gamma frequency oscillation during the stimulus presentation, sort of a bottom-up presentation of the stimulus, and then beta bursts of activity during the delay maintenance. And we can talk about that. The brief answer to that, or my brief criticism for that, is that we haven't seen compelling evidence that these models account for behavior better. They don't really explain something about the pattern of errors that subjects do, or provide more information beyond these firing rate models can do. Okay. All right, very well. So we now armed now with this understanding of this framework, basically, let's move on to the main focus of our research here, which is deep brain stimulation targeting the nucleus basalis of Maynard. And we did this experiment with an eye towards translational applications, perhaps using this as an intervention to improve a working memory and cognitive function in aging. Okay. So let me illustrate how we did these experiments. So we implanted chronically, permanently in the brain an electrode that reached deep into the basal forebrain, the nucleus basalis of Maynard. This is the approximate location, basically, of the electrode. And this electrode stays there, but we also had the ability to place a recording cylinder in the prefrontal cortex. And at the same time that we do these stimulation experiments, we can also record neural activity from the prefrontal cortex of these monkeys, so that we can have not only the behavior, but also see how the activity of individual neurons in the prefrontal cortex is affected. This is the histology to show exactly where the tip of the electrode was, and we verified that we're in the right place by immunohistochemistry staining for neurons that produce acetylcholine with anti-chat antibodies. Now, the stimulation parameters turn out to be non-trivial. How do you stimulate the nucleus basalis to improve the release of acetylcholine to improve cognitive function? So generally, in all of the experiments, we use this kind of biphasic pulses. We use 200 microampere cathode first pulses that we repeated every 12.5 milliseconds or every, this is variable. This time, as I will show you, is variable. Basically, if we place this train of pulses at 12.5 milliseconds, that will generate an 80 hertz frequency of stimulation, and the step of each pulse is 100 microseconds. So that's our sort of our basic stimulation parameters. We can model what is the spread of activity, what is the expected spread of activity if you generate something like that. And the spread of activity is in the order of 1 millimeter. So the reason that, again, we got into the, went into the trouble to do these very invasive experiments and use this deep brain stimulation electrode is because they allow us very precise activation of a really tiny piece of brain, basically, unlike some less invasive methods, basically, such as focused ultrasound or other or transcranial magnetic stimulation, which I have nothing against. These are evolving and they're improving, and I'm certain that there will come a time, basically, that they can produce a great stimulation. But for us to start with, we relied on this deep brain stimulation precisely because we have very good control of the spread of stimulation, and we can activate just a tiny piece of cortical volume. All right, so when we first thought of this experiment was, we're going to put this electrode there, we're going to blast the nucleus basalis with 80 hertz continuous stimulation. People in movement disorders use 120, 130 hertz stimulation, basically, to drive the basal ganglia. So let's start from that, and let's see if that improves cognition. And as it turns out, not only did it not improve, but quite dramatically and quite spectacularly depressed, working memory performance. We had, this is the control, and this is with this 80 hertz stimulation parameters. We had two different monkeys. Their performance went down the drain when we applied that. So we had to go back and think about it, basically, and think, what are the physiological parameters of stimulation, how neurons in the nucleus basalis fire, and these are not super high firing neurons. Therefore, we figured out that this regime of very high frequency continuous stimulation somehow must be depleting acetylcholine or inhibiting. The net effect must be some inhibitory effect, basically, or the neurons cannot be entrained at such high frequencies. So after a few tries, basically, we came up with this protocol of 60 hertz intermittent stimulation. So we generate a 60 hertz train of pulses. However, it's not continuous. So we do it for 15 seconds, and then we pause for 45 seconds, and then repeat the cycle every minute. These are different parameters here to avoid ceiling effect. But now, when we compare control and stimulation conditions, we saw very consistent increase in performance in working memory tasks. And across the entire curve, basically using different delay intervals, very consistently, we saw that this pattern of intermittent stimulation can improve working memory. And we have replicated this finding now in three different institutions, essentially 3 different labs, and with different tasks. So we are very confident that this is a robust finding, and this regime of stimulation works, and it does improve behavior. All right, the way we did these experiments allowed us, as I described, to not only record behavior, but also record neural activity. And let me briefly explain exactly how we did this experiment. First of all, the task that we used here was a variant of the delayed response task, but it was a little more difficult, a little more challenging, and in fact involved presentation of 2 stimuli in every trial. So there's one stimulus that the monkey is seeing, and then there's a second stimulus. And depending on the color of the fixation point, being either white or blue, the monkey has to remember either the first stimulus or the second stimulus. So this is a little more difficult task that allows us to challenge the monkeys more and just tease apart some important parameters here of the experiment. So this is sort of a schematic diagram of one trial. There's a fixation point here. The first stimulus is presented. There's a delay period. Then the second stimulus is presented, second delay, and then the eye movement. So we can have one trial take something in the order of 6 or 7 seconds. Then there's an intertrial between trials. And we can now then structure our task where we have 4 or 5 of those trials span an interval of about 45 seconds. And then during the intertrial interval, we apply our deep brain stimulation. So our deep brain stimulation is not tied to the stimulus presentation, or is not tied to the delay. We just, we are able to sort of increase or decrease this level of arousal or of cognitive basically focus, if you will, basically by repeating this stimulation every minute for 15 seconds. Okay, and we also have some conditions where the trials are structured exactly the same way, but there's no stimulation. There's just a long intertrial interval. Otherwise, the monkeys, obviously will tell apart that something is different, basically in different types of trials. Okay. All right, and this is then what prefrontal neurons do under nucleus basalis stimulation. Okay, this is again the x-axis here is time. This is the presentation of the first stimulus, first delay period, second stimulus, second delay period, and this vertical line is their response. So overall, stimulation of the nucleus basalis improved or increased firing rate of these neurons. This is just one example neuron here, but we saw that very consistently. However, again, unexpectedly, we saw a result that we're not expecting at all. So if we plotted now that tuning curve of individual neurons and the population as a whole, the blue line is the control, the orange line is the stimulation condition. We saw that, yes, the activity overall has increased. However, the tuning of neurons has broadened, has flattened. And this was true both for the stimulus presentation period and for the delay period. And this was a result that, we really struggled with. Excuse me for a second. I did my postdoctoral training with Patricia Goldmarquez, and I was there in the lab when she was doing these, wonderful foundational experiments of dopamine microendophorosis to tease apart the effects of dopamine in working memory. And that the result of this experiment basically is that under certain concentration conditions, neurons would sharpen the tuning and improve the tuning, or neurons that appeared flat on baseline will become tuned. And this has been replicated with acetylcholine as well. So low dose of acetylcholine agonists generally tend to sharpen tuning of neurons. However, if there's a high dose of cholinergic agonist, these will tend to broaden the tuning. These are experiments that have been done in primates with microentophoresis. So you put a tiny syringe, tiny needle in the brain, and you squirt a tiny quantity of agonist, and you can see the effect of neurons very locally near the tip of this injection. However, these experiments were done independently with behavior. It is assumed that for these low-dose agonists, basically, the performance of the monkey goes up, and the sort of high dose of acetylcholine agonist corresponds to a descending curve, to a descending limb of this performance curve, where if you give too much agonist, then you degrade performance. However, I'm saying again that these two experiments were done independently. It's not possible. Microentophoresis by itself infuses such a small quantity of this drug that generally is not sufficient to produce any change in behavior whatsoever. So we have come to question that in one of our future experiments. One of the things that we want to do is revisit some of these drug experiments, basically, and find out exactly what is it that happens in the activity of neurons when you give the animal basically a cholinergic agonist that that improves or degrades performance, because our nucleus basalis basically results seem to indicate that this model is not true. All right. Then the second question we had is like, how is that possible? How can you have this sort of flattening of tuning of neurons and improve performance? So we went back to our bump attractor model and tried to understand, tried to simulate, what is the effect of a wider bump, basically. And I'm showing here first the qualitative description of that, so the intuitive description of what the bump attractor predicts under these conditions. So on the left, I'm showing a trial of our task that we use that involves these two stimulus presentations in the off condition, the control condition. So when you present a stimulus, that will generate a bump of activity somewhere in the network, and we present a second one, basically, we know from other experiments that we get a transient activation, but generally this bump returns to the original position, and this activity of bump would drift exactly as I described. We expect to see some random drift from trial to trial that causes these inaccuracies that we see experimentally. Now, with a stimulation on, if the bump is wider, generally would expect that this drift will be less. So this system, you can think of it as having as more inertia, is more stable. So since more neurons are active and you have this wider bump, basically, when the stimulus is on, this will just make it less susceptible to this random drift, basically, and therefore would expect the accuracy of the system under the regime where neurons have wider, broader tuning curve to be better. However, there's a trade-off, and that is there are some special cases where the stimulation can actually have a detrimental effect. So if we place the second stimulus presentation close enough to the first, this is the first and this is the second now, there comes a point where this pattern of activation, basically of the second stimulus, would tend to merge with the first, and then you're going to get an error, basically, because the two bumps are going to merge closer to the location of the second stimulus. And when we went back now to the behavioral results of the experiments, and we saw, these are the experimental results, and we saw what happens as a function of distance between the two stimuli, we in fact found out that despite the overall improvement in working memory, for this condition where the two stimuli are close, then the system loses accuracy and the performance actually declines. And the second thing that we saw is precisely that the pattern of errors, the distribution of endpoints was much tighter under stimulation, was much closer to each other under stimulation than under the control of the same animals in different blocks of trials, basically. All right. So this sort of qualitatively, at least, provides us with some kind of an understanding of how is it possible to broaden the tuning of neurons and improve at least some aspects of behavior. Most recently, in collaboration with Albert Comte again, we saw how it's possible for this to happen with acetylcholine. So we know that the effect of acetylcholine is generally facilitatory. So it increases feed-forward connections. And this is sort of the analysis and the explanation of how it's possible to get this broadening of tuning and how this can actually improve behavior. All right, so first of all, it's important to appreciate that the system behaves very differently under two different conditions. If it's non-saturating input-output, if any increase in feed-forward excitation basically produces higher and higher and higher firing rate, then when you apply acetylcholine with nucleus bisalazone, then in fact you would expect to see this higher firing rate only for the peak of activity, and you would expect to see this sharper tuning as the classical models would predict. And again, clicking here. However, real neurons are not like that. Neurons saturate, and then the modeling showed basically that if you have saturating neurons, then by facilitating this feed-forward input, as acetylcholine is presumably doing, can in fact produce this DC shift and this broadening of tuning, basically just as we saw experimentally. And how could that improve behavior? So we can again model this what Albert called the diffusivity. How much drift we have from trial to trial. So we can have a metric basically of the dispersion of the endpoint of these trajectories and find out how that basically changes with this feed-forward input. And in fact, the model predicts that the diffusivity will be less if you have this broader tuning. Basically, if you have this more stable attractor, then this diffusivity will be less. So this result, gave us for the first time some mechanistic explanation of how an agent, a drug basically, that generally has facilitatory effects and increases feed-forward excitation can in fact cause this broadening of tuning and at the same time account for improved behavior. Now, all of this modeling, the previous slide that I saw, were dependent on the bomb attractor model, on this ring model that I showed you, which is a very simple model. All the neurons have identical properties. It makes a lot of unrealistic assumptions. So it was important to replicate these results using a sort of more sophisticated model, and we used the heterogeneous spiking model that was developed by David Hansel and Matto, and this, essentially behaved in the same way. In all the factors that matter for us, basically, as this feed-forward input increased, this diffusivity generally decreased, and this model also produced this effect of higher, basically, of less tuning. And when we modeled independently different aspects, basically, or different changes that could be attributed to the effect of acetylcholine, such as increase in feed-forward input, change in the synaptic weight, etc., the critical combination, or the changes in the network, basically, that accounted best for the experimental results with this increase in feed-forward input, but at the same time, a decrease in correlations, in neural correlations between neurons. And this is something that we have not tested experimentally in these experiments that we did, because recordings are difficult, when you have a lot of instruments basically in the monkey's head. We didn't have large numbers of neurons recorded simultaneously, but this is something that I want to do, that the model clearly makes this strong prediction that correlation between neuron decreases. So spike count correlation basically would expected to be decreased under the effect of acetylcholine. So this is where we stand with our understanding of the effects of nucleus basalis stimulation. I just want to end here my talk with what we're planning for the future. So, and as I alluded, we really think we have found an intervention that is effective in improving working memory and cognitive function in young, adult, perfectly healthy monkeys. So this obviously has much more values for individuals that have impairments of some sort. So we're very interested to see if we can do the same, have the same effect and produce the same benefits for aged monkeys. And we did this experiment by now implanting implantable pulse generator. This is the real FDA-approved IPG that people use for movement disorders, for Parkinson's disease. Typically in humans, you would implant it in the chest cavity. We didn't want the monkey to try to pick it, so we implanted it in their back, basically, and we ran a wire directly under the skin of the skull, so we can have an electrode basically targeting the nucleus basalis. And then these things are wireless, so we didn't even need to connect any connectors. So the monkeys could now do their task in their cages while we turn this on and off for 1 hour a day and deliver stimulation. And we could, very robustly determine performance by using an adaptive tracking procedure of how long a delay period the monkey can do with having to remember, a stimulus basically here, and there's a delay period, and they have to choose one of the two. And then we use this estimated threshold then as a quantity, as a measure basically of cognitive performance under stimulation and without. We've tried that now in 8 different animals, and obviously there's a lot of variability within each animal, but the effect was highly significant, and it definitely improved working memory performance in these aged monkeys. These monkeys we had some monkeys that were about 25 to 30 years old, and generally monkeys age at a rate of 3 to 1. So, these are really old animals, geriatric animals. That's how we affectionately refer to them in the lab, basically. And we saw that all of a sudden they can have an epiphany and they can improve performance. The other thing that we're interested in, and the first part of my talk basically described experiments that were acute. We had a stimulation that would turn it on, and we recorded from neurons and we turn it off basically. But this experiment didn't really address how long the effectiveness of this intervention is. We know that acetylcholine drugs, if you're diagnosed with Alzheimer's disease, you immediately start an Aricept. You immediately start with a acetylcholinesterase inhibitor, which has effect. However, this completely was out in a matter of 6 months. Within 6 months, patients have returned basically at the baseline and below that because the disease progresses. So for us, it was important to track these animals for several months, and we did that for up to 15 or 16 months. And the red curves here represent performance as a function of 100% of baseline for those animals that did have this intervention, and the black lines are controlled that were either were not implanted or the stimulation was not on. And we in fact saw that the stimulation lasted for a long period of time, definitely more than 6 months. So we know that we believe that this is viable, and we're now planning a human clinical trial that we can use deep brain stimulation targeting the nucleus basalis of Maynard as a potential therapy for Alzheimer's disease. So that's what I wanted to discuss. I will stop here and we can have questions and discussion, but let me summarize the findings then of my second part of my talk. So I saw that intermittent nucleus basalis stimulation improves working memory. And I underline that because if you blast the nucleus basalis with continuous stimulation, that's not going to have the desired effect. I saw that quite unexpectedly for us, the nucleus basalis stimulation increases firing rate However, it broadens the tuning of prefrontal neurons. We saw that we can explain that in terms of a more stable attractor. So we expect to see a behavior improvement because the attractor now is more stable when these neurons have this broader tuning. And I showed you that stimulation is effective in aged animals, which opens the road for us for potential interventions. I want to thank the people who did this experiment. These are my lab members who participate in these experiments. And on the right, these are my collaborators. I mentioned Albert Comte's name several times, so he was part of all of these experiments. Dario Englot is the neurosurgeon who assisted us in a lot of these animal surgeries, and he's the lead person now in this future human clinical trial. Dave Blake is a biomedical engineer and colleague of ours who had a big part in the development of the stimulation parameters. And Shuyan Chi is my former trainee who did these prefrontal recordings. So I'm going to stop here, and I'll be happy to take questions and discuss further any topics you'd like to discuss.